This lecture will cover the development of the heart. In it, we want you to be able to start with the primitive heart tube and then describe the movements that occur as the heart tube converts into the primitive four-chambered heart. From there, we'll discuss atrial and ventricular septation, and you'll be able to understand how interatrial and interventricular defects can occur. And these defects in the cardiovascular system are perhaps the most common congenital defects that are seen. We'll talk about interatrial defects and compare them to pulmonary stenosis of the right ventricle. And then in the next lecture, we'll talk about the development of the great vessels and the changes that occur as the fetus is born and the embryo starts surviving on its own. To discuss the development, we'll start with a horseshoe-shaped pericardial cavity. We've already seen this in introductory lectures. We'll talk about how that horseshoe-shaped cavity via lateral body folding becomes a single heart tube. We'll talk about the convolution of that heart tube, the formation of the primitive four-chambered heart, and atrial and ventricular septation. It's about day 18, and the embryo begins forming blood islands. Blood islands are groups of cells called hemangioblasts, and they will form the prospective myoblasts as well as the prospective angioblasts that will go on to form the blood-forming cells. The blood vessels that are forming from these hemangioblasts will initially form an endocardial heart tube, and it's a crescent-shaped structure in a precephalic location. That is, it's in front of the developing brain and in front of the oral plate. As a result of lateral body folding, what we can see is that the heart, which is developing in the splanchnic mesoderm, is going to be moved in a medial direction, and as it moves in a medial direction, it is going to come to lie in front of the foregut as the foregut is pinched off from the vitelline duct. If we look at the embryo and sagittal section, what we can see is there's the oral plate, and in front of that we have these blood islands, and these blood islands then are going to rotate both ventrally and caudally as a result of the increase in the development of the brain. So as the brain develops in a cephalic direction, you'll notice it moves the oral plate and the heart tube caudally. That continues until the heart is in pretty much what would be the equivalent of the cervical region of the embryo. So if we look at the development of the embryo again and look at the cross-section and look from below, we can see that the horseshoe-shaped pericardial cavity is going to be cut in cross-section here, and that these two endocardial heart tubes are going to move towards each other, and the two endocardial heart tubes are going to fuse. They're going to fuse at about day 21. And the significance of the fusion is that when they do, the heart begins to beat. The first region that fuses is in the ventricular region. So here we see those two heart tubes surrounded by cardiac jelly. This is surrounded by a myocardial mantle. This is the future heart, and these two tubes will fuse into a single heart tube. Notice that the heart is in front of the foregut. There's the foregut, there's the heart, and it's attached to the posterior body wall by a dorsal mesocardium. As you remember from gross anatomy, that dorsal mesocardium is going to disappear and only be attached at the inflow and outflow ends of the heart. So now we have a heart tube sitting in the body cavity, and the next event that's going to occur is that we're going to get that heart tube convoluting. One could ask, what are the signals for the heart tube to form? Here we're looking down at the embryo, and if we look down at the embryo, we can begin to see that there's differential expression of different genes. GATA4 is probably the most important gene for the beginning of cardiogenesis. In addition, the endoderm is going to secrete BMP2 and 4, and that BMP expression is going to be important in the formation of the heart tube. Crescent and cerebrus are going to be important in inhibiting the Wnt genes, and the combination of the BMP secretion and the Wnt inhibitors is going to allow for an upregulation of NKS 2.5. NKS is very important in terms of the development of the cardiovascular system. In addition, fibroblast growth factor is important for some of the heart-specific gene proteins to be expressed. So now we have our heart tube. We have an atria, a ventricle, have an outflow region, and the outflow will go to what are called the aortic arches, and we have an inflow region, and that will receive blood from cardinal veins, from umbilical veins right here, and from vitelline veins. So the venous drainage will be into an inflow region, which takes the blood to the atria, to the ventricles, out the aortic arches, and down to the arterial system. Now, if we look at that heart tube, we can begin to identify via some bulges that occur several different regions. As I said earlier, NKX 2.5 is important for this differential development of different regions of the heart tube. If we look at the inflow region, which is developing caudally, we have two sinal horns. Here we see now the atria have fused, the primitive ventricle is fused, and we have a bulbous cordus region. That's the outflow region. The bulbous cordus can be separated into a truncus, more caudally, and a conus region more, a truncus region more cephalically, and a conus region more caudally. These will then continue with an aortic sac, which then goes and distributes blood to the aortic arches. In terms of the development of these different regions, it's been found that there are different genes that are expressed in different regions, so that in the conus and the left ventricle, hand 1 is important, and hand 2 is important for the development of the right ventricle. TBX5 is important, that specifies the atria. So several things to be noted here. One, there are differential gene expression that allows for these different regions to develop into the different parts of the heart. And in addition, we need to note specifically that the right ventricular cells are different, and they come from a different source than the cells of the left ventricle. So you would expect that the right and left ventricle are different. In addition, retinoic acid is important. It comes from the mesoderm, and then the atria and the sinus venosus will produce retinoic acid. And what we see is that the higher levels of retinoic acid are down at the inflow area, with lower levels in the outflow region. When we think about retinoic acid or vitamin A, you can, with excess vitamin A being taken, get congenital abnormalities, and infants can demonstrate small, abnormally shaped ears, lack of growth of the mandible, cleft palate or palatal defects, as well as congenital heart defects. So an excess of vitamin A can lead to some defects in heart development. Once again, we want to point out that the vitelline veins, the umbilical veins, and not shown here, the cardinal veins, all will empty into the two sinal horns. The next thing that's going to happen is looping. And again, looping depends on laterality inducing genes, that is, there are right-hand genes and left-hand genes. And that will cause convolution of the heart tube. So if we look at the convolution of the heart tube, what we're going to see is there will be a bulboventricular loop that forms and an atrioventricular loop. The bulboventricular loop forms between the bulbous cordus and the ventricle, and that loop goes forward or ventrally and caudally. The atrioventricular loop is going to go posteriorly and cephalically, so that we end up with the two atria posteriorly located, two ventricles more anteriorly located, and you see now sort of the basic organization of the heart that you saw in gross anatomy. If we look at this in a sagittal section, this developing heart, this primitive four-chambered heart, what we would see is the following. You would see the atria posteriorly and cephalically, then a ventricle. This is still the common ventricle or primitive ventricle, which would then have the blood flowing to the conus region and the truncus region. There would be an atrioventricular canal and a bulboventricular canal. If we look from the front then and remove just this part of the developing primitive four-chambered heart, we would see the following. We would see the two atria posteriorly. There would be the AV canal, which is going to open into the primitive ventricle initially, and then the primitive ventricle is going to be continuous with the conus region and then the truncus region. Now we know that this atrioventricular canal is going to have to move over here to the right, and there'll have to be a regression of this tissue here called the bulboventricular flange. Again, if we look at a sagittal section of the primitive four-chambered heart, this is what we would see: atria, ventricles, and the truncus and conus. And now we can make the point that in the atrioventricular region we'll begin to get a growth in the center of the AV canal. So if we look in a cross-section here of the developing heart, not quite a cross-section, but pretty close, where we cut through the atria and the ventricles and look from the front, what we would see is the following. The two atria, two ventricles, and the common AV canal. The right ventricle developed from the conus region. The left ventricle developed from the primitive ventricle. And the remnant of the bulboventricular loop is here, where we're beginning to see a little bit of the interventricular septum forming. The atrioventricular loop is here. And so basically that AV canal has started migrating to the left. Excuse me, to the right. Here we see the two atria. And this slide shows that the AV canal is going to shift to the right, and as it does so, this bulboventricular phalange is going to regress. This allows the right atrium to have blood flowing through the AV canal into the right ventricle, and the left atrium having blood flow through the AV canal into the left ventricle. This cartoon just depicts that growth of cells into the AV canal that we talked about, where we begin to get the formation of endocardial cushions. The endocardial cushions are going to fuse in the midline, and they will form this endocardial cushion in the center of the AV canal, thus forming a right AV canal and a left AV canal. Now, as that happens, we begin to see the heart undergoing atrial septation. The first septum that forms is called septum primum, and it will grow downward towards the endocardial cushion. Initially, it leaves an opening between the septum primum and the endocardial cushion. And that opening is called the ostium primum. So we have septum primum, ostium primum, the endocardial cushion. Now, because of the fact that the blood eventually is all going to empty into the right atrium, and because we need blood going from the right atrium to the left atrium for the fetus, this opening has to be maintained. That is to say, we need to have a shunt from the right atrium to the left atrium. But ostium primum is going to close, and so basically a second opening has to develop. Here you can see the development of septum primum and ostium primum beginning to close. And here you can see the beginning of a second foramen or ostium opening in septum primum. And this occurs because this region undergoes apoptosis, or programmed cell death. As ostium primum fuses with the endocardial cushion, ostium secundum opens, and we see an opening from the right atrium to the left atrium. Thus, blood from placenta will travel via the ostium secundum from right atrium to left atrium. The next thing that's going to happen in atrial septation is that we're going to get another septum forming. It's the second septum, and we'll call it septum secundum. That's going to leave an oval-shaped foramen to the right of the septum primum. In addition, there'll be two small septa on either side of the inflow of blood from the sinus venosus. The sinus venosus is actually going to become incorporated into the right atrium and help form a little bit of the septum secundum. And this is going to become the smooth-walled portion of the right atrium. And that comes from the sinus venosus tissue. So all the blood is going to empty into the right atrium. It is going to go through an oval-shaped foramen in between the septum secundum and the endocardial cushion called foramen ovale. The blood is then going to go across through ostium secundum into the left ventricle. Right atrial pressures are higher than left atrial pressures in the fetus, and hence the septum primum can act as a valve. and prevent blood from flowing backwards from left to right. And in fact, we will call the septum primum, the valve of the foramen ovale, which is located here. You saw this tissue in the gross anatomy lab as the fossa ovalis when you looked at the inner atrial septum. So now we have the formation of foramen ovale, the septum secundum, and the septum primum, now called the valve of the foramen ovale. And what is going to happen is at birth, now these left atrial pressures are going to elevate, becoming higher than right atrial pressures, and will force septum primum against septum secundum, and we'll get the formation of the inner atrial septum. So now we can see the atria formed, the superior vena cava and inferior vena cava emptying into the right atrium, the blood going from there through foramen ovale, through the ostium secundum into the left ventricle. The coronary sinus would also be located in this particular region. In terms of the left atrium begins to form as a primitive atrium, and then we'll have the formation of the pulmonary veins. Initially, it's a single vein that becomes incorporated into the left atrium. This pulmonary vein is going to develop and bifurcate, and that tissue is going to be absorbed into the left atrium up to that first bifurcation. So we'll have two pulmonary veins emptying into the left atrium. These vessels will bifurcate as well, and they will become incorporated into the left atrium. So most of the smooth-walled left atrium that you saw was developed from pulmonary vein tissue, which you can see here. So now we have the four pulmonary veins emptying into the left atrium, and over here the superior vena cava, inferior vena cava, and not shown the coronary sinus opening into the right atrium. So during the fetal life, blood can continue to flow from right to left. And this slide just shows you the pulmonary vein tissue forming much of the left atrium, except for the left auricular appendage, and the sinus venosus tissue forming that smooth wall portion of the right atrium, right up to the region of the crista terminalis. The rest of the right atrium is from the primitive right atrium tissue, all the pectinate muscle and the right auricular appendage. In terms of the interventricular septum and how that forms, that's going to form initially from a growth in the region of the bovoventricular loop, and we're going to form a muscular interventricular septum. But we do want to remember high up we have this membranous septum, and that's going to form from three components: the septum membranaceum. So if we talk about ventricular septation, we see the bovoventricular region here and the bovoventricular canal, and we begin to see a growth in this particular region of the muscular interventricular septum, and it will separate the right ventricle and the left ventricle. That interventricular septum is going to grow towards the endocardial cushion. Initially, there will be a very large interventricular foramen. That interventricular foramen is going to get smaller as the interventricular septum approaches the endocardial cushion, and will then form what is called a secondary interventricular foramen. This secondary interventricular foramen is going to be closed off from three sources. Those three sources are a muscular interventricular septal tissue, that is connective tissue from that muscular septum, from endocardial cushion tissue and from ridges that spiral down from the septation of the truncus and the conus and separate the pulmonary and aorta. So you can see the ventricular septation and the formation of the septum membranaceum is going to come from the connective tissue from the muscular septum, from endocardial cushion tissue, and then we need to fill out the rest of this, but to do that we need to look at the heart as we take here a cross section and look from the right. And as we take that cross section, look from the right, sagittal section, look from the right, this would be a sagittal section of the heart going right through the muscular septum and the interatrial septum. Here you can see the inferior vena cava, superior vena cava, coronary sinus as we describe them emptying into the right atrium. Here you can see in the interatrial septum the foramen ovale. As we go down to the base of the atria, you can see the endocardial cushion that is formed in the midline. The endocardial cushion, the muscular interventricular septum contribute to the closing of the interventricular foramen. And the final component that closes that interventricular foramen is from what is called the bulbar septum, the conal septum, if you just want to think about the lower part of this, or another term is the aortical pulmonary septum that separates the aorta and pulmonary artery. This septum I'll describe in more detail in the next lecture, and we can see that it will spiral down and complete the closure of the interventricular foramen and form the septum membranaceum. So these three components then contribute to the membranous septum, that is the bulbar septum, the interventricular septum, and the endocardial cushion tissue. They will all form the membranous interventricular septum.